next period. I do that at lunch. No, she's going to remember. You're going to remember? I'll feel like a complete jerk. That was very unintentionally rude of me. Okay, anyway. I talk too much. You may have noticed. Okay. Today we're going to talk about another important class of biological molecules, and this is the amino acids. Have you ever heard this term before? Yeah?
Yeah, I have.
And in what context?
Nitrogen.
Oh, okay, interesting. Cool. Nice. So amine, amino, right? So that clicks back to the functional groups we learned. I like that. And even better, he knew which one it was. Amino functional group is the one with the N, nitrogen. Good. Has anyone heard it like in the day-to-day lives?
Yeah, like nutrient-wise, I hear a lot like using amino acids and like certain drinks you can drink.
Yeah. So when people are trying to sell you expensive food, possibly overpriced food, the science words start flying. Amino acids. Ooh, fancy. Can't get those anywhere else. But yeah, amino acids. We think about them in terms of health. And we're actually going to talk about that because there's nothing to that. Well, yeah, we'll give it a minute. So let's break the name down and go back to our functional groups. Amino acid. Remind me, what we just looked at it in the case of chitin, which had these N groups in it. And we said that the amine group confers what property?
Basic.
Makes it basic. And yet, this is an amino acid? Qu'est-ce que c'est?
I don't know French.
Qu'est-ce que la hell, as my high school French teacher used to say all the time. But how's that work? If we look at our functional groups, we saw that amine groups are basic. And yet we're told here this is an amino acid. What might we be looking at here? What do the acids look like? Which functional group confers the property of acidity? A carboxyl, which itself is almost like an amalgam of two functional groups. What are the two functional groups on a carboxyl or a carboxylic acid?
Hydroxyl and carbonyl.
Yeah. So lots going on there. And yet the very name is going to tell us that this thing, an amino acid, must have one carbon on it that has an amino group, and it must have another carbon on it that has a carboxylic acid, a carboxyl group. Does that make sense? And indeed, that's exactly what it is. So it's well named. It's well named. The general form of an amino acid is this. We have a central carbon, which is making one bond to hydrogen, and is making one bond to an amino group, which means what? And typically we put this on the left when we draw it, but sometimes we'll put it on the right. Try and stop me. What's that going to look like? Nope. Amino. N. And how many bonds does nitrogen want to make? Okay, and this chain ends. This doesn't keep going, so what must be on those two bonds with N? Yeah, hydrogens. I mean, I guess it could be fluorine, chlorine, or bromine, or iodine, but those impart very different chemical properties, which aren't present here. So that takes care of the amino portion of it. How do I take care of the acid portion of it? Where does that go? Could I put it on this carbon? Could I go like that?
Maybe.
I could. The only problem is, if this is a functional group, what else does it have to do?
Function.
Don't we all? Well, it has to attach to whatever you're attaching it to. As this is drawn, can it attach to anything you're attaching it to? No. So the carboxylic acid can't be on that one. So what do we need? Another carbon. And this is a carboxylic acid. And quickly remind me, what's a carboxylic acid or a carboxyl group?
Double bond to oxygen and a bond to OH.
Very good. Just like that.
Are all these saying O negative?
Well, because as an acid, when it dissolves in water, the whole point of an acid is what does this hydrogen do? It pops off. That's what makes it an acid. So yeah, you'll sometimes see this written as O negative, and that just means it's been dissolved in water. This would be its crystallized form. And then, where is this going to lob on? Yeah. And there's your R group. That's enough for today? No, I don't think so. We're going to keep going. So this is different on every different amino acid. And you could put just a hydrogen there. You could put a carbon with 3 hydrogens there, or you could put something else there. And each one will have its own unique properties. You could put a functional group there. You could put a hydroxide there. You could put a second carboxylic acid. You could put a little ring there. You could put whatever you want. And in practice, amino acids are defined by the different R groups that can be placed as the 4th bond on their central carbon. And while there's potentially an infinite number of amino acids that could be made, right? You could put anything there. In practice, in your body right now, there are exactly 20.
What if I have 21?
You would. And that last one, oh, it's a good one. So if you look here, yeah, Preston noted that they show this as NH3 and O-. That's showing you the acidity and basic property of this. That's showing you that this oxygen has acted as an acid by popping that hydrogen off, like Preston said, and then this NH2 has acted like a base by accepting it. You don't have to worry about that, but that's all that means. Does that make sense? So it's an acid on the right side and a base on the left. Interesting. That gives it interesting behaviors. It also means that this molecule isn't even just polar, it's actually charged. So it's very, very, very hydrophilic. Plays nice with water. So if you look here, there are 20 different side chains that can be attached. And with the 20 different side chains, you get glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, proline, serine, theanine, cysteine, tyrosine, asparagine, glutamine, aspartate, glutamate, lysine, arginine, and histidine. At one point in my life, I could draw all of these from memory. That time has long since passed. But there was a time when I had to memorize all of them. It was not a fun time. Yeah.
It's weird. The only one I've ever heard, like on TV or something, is glutamate, and maybe aspartate. But those are the only
You've
two.
seen this somewhere.
Probably maybe. I don't know. All I remember is glutamate.
Yeah. But
Asparagus.
But if you look here, they are kind of categorized in this chart by what property? What property are they categorized by in this chart? They've been grouped together. How have they been grouped together? By one of the major chemical properties something can have.
I don't know.
Polarity. So we're told that all these ones here, the side chain is nonpolar. No charges and no... What functional group would we expect to see in these side chains if they're going to be polar? What functional group makes things polar? Sorry? Hydroxide groups, right? Hydroxyls. And do we have any in any of these side chains? So those are the nonpolar side chains. And then after that, we get the polar side chains. And what do you see in every single one of these? Well, you kind of do, because sulfur obviously is different than oxygen, and yet look at sulfur on the periodic table, and it is isoelectric with oxygen. It behaves like oxygen, right? It's right underneath oxygen. It also likes to make 2 bonds, and that is also a polar bond. So every one of these has a polar side chain. These do it a little differently too, which, yeah, we'll get to.
With another amino acid?
Not an amino acid. It's different because they're coming from the same carbon, which is its own functional group. called an amide. Don't worry about it. Don't need to know it. And then the last group actually have not only polarity, but they actually have full-on charges, either a positive charge or a negative charge. Which of these will be hydrophilic? Which of these will like water, interact well with water? The polar ones, sure. What about the electrically charged? Will they interact well with water, which is polar? Yeah, the whole point of polar interactions is that they're these partial charges, right? And the partial positive of one water is attracted to the partial negative of the other. Well, all the better if you have a full charge. So all of these have hydrophilic side chains. 1L, right? This is what we determined. Thank you.
No why.
I apologize. Thank you. I apologize that I'm supposed to be leading you educationally and I can't even spell the words I'm supposed to teach. And the non-polar side chains will result in these being hydrophobic.
Also no why.
But note that that's not the only property that these things have. They also have properties such as different sizes, right? Just different shapes, which is important. We'll get to that. They have different chemical interactions. They will react with different things. They will react with each other differently. So polarity is one piece of this. Hydrophobicity and hydrophilicity are a piece of this, but they're by no means the only part of the puzzle.
What's with the fancy water bottle?
It's my daughter's. I stole it.
Makes sense.
I mean, I didn't steal it permanently.
It's very glittery.
It's nice, right? I shouldn't even say I stole it. Yesterday on the way out of the door, I said, "Oh, God, Claire, could you make me a cup of tea?" And she made me a tea, which is nice. But I didn't bring it back yesterday, which if you're wondering if she was pleased about, she was not. She was displeased.
So you're still going to use it?
The difference is if I use it or not. Yes. Nice cup of tea. I think it's a good color. I like it. Okay, so
It's shiny.
as you can see, and sorry, this here should have been on the previous page. Something got weird here. As I ported this to my new font set this year, it got weird. That should be on the previous page. But as you can see, the side chains, the R groups, have their own functional groups, and they have their own chemical properties. This will be important when amino acids are put to use. And how do they get put to use? What do amino acids make? Yep, and what is the polymer of an amino acid called? Proteins. Very good. Amino acids make proteins. And they are formed by another condensation reaction. In this case, we call it the peptide bond. So peptide as a prefix always refers to proteins. In fact, more specifically, amino acids, but that's okay. Just in terms of, as a reminder to yourself, anytime you see the word peptide, we're talking about proteins. Anytime you see saccharide, we're talking about sugars. Anytime you see peptide, we're talking about proteins. So let's draw a peptide bond. Obviously, how many amino acids are we going to need to show how, at a minimum, to show how them bonding together would work? Well, at least 2, right, if they're going to bond. And by convention, we draw the amino group on the left, and we draw the carboxylic acid on the right. As much as anything, that's because it matches the name. What 2 other things does this carbon bond to? A hydrogen up above and down below. Sorry? Yeah, the R group. So it could be anything. Whatever side. Oh, hello. Wow. Whatever that side chain is. And let's take a second one. And maybe you can predict how this is going to work. So here's my second amino acid with carbon in the middle. And that there. And I wish I'd left a bit more space because I'm going to have to draw the other half of this reaction. So I'm going to move it over. If you've gone too big, I'm sorry.
Oh.
You could always just highlight it and slide it over.
You give us a forewarning.
I'm so well. If I had a forewarning, I wouldn't have drawn it in the wrong place to begin with, Preston.
Why don't you look at your
I don't
lesson?
plan
You
my
made it.
I don't plan my failures. I am looking. It fit very nicely on this one. Leave me alone. Leave me alone. I'm not being mean to you. I'm gonna go sit here with Ryan. Ryan's never mean to me. Actually, sometimes Ryan's mean to me, but usually very quietly under his breath. So it's different. You can call me out publicly like
Yeah, but now
Preston.
you're sitting beside
I'd shut up. Nobody asked you. So, how did these come together? Take a look. What's it got to be? How did the sugars come together? What did they make? What got released that allowed them to bond? Yeah. Sorry. Okay. Which H?
On OH.
Yeah. So the OH is going to be involved. That's one of the attack sites. That's what we call it in organic chemistry. And what's it going to react with? No. Because the problem with that is, if that was the way it worked, could you ever make a chain? No, you'd only ever get two, because there's only an OH on one side. The nice thing is, N isn't another carbon. Nitrogen, a bit more electronegative, which means both of these hydrogens are kind of up for grabs. Does that make sense? A carbon bound to a hydrogen, not really up to, the hydrogen isn't up to for grabs. Carbon's just too neutral a bond. But nitrogen is kind of electronegative and is happy to kind of play there. And so this becomes up for grabs, and that right there is what's going to form. So what do they release? What is that?
Water.
That's water. All right, so that's easy. And then we have the first amino acid again, H, N, H, C, H, R1. I meant to label the next one R2 because, of course, they don't have to be the same, though they could be. And now, this carbon is directly connected to the next one. And just to keep the shape from getting unwieldy, we usually will draw the double bond to O straight up. And then this bond here that used to be to an OH is now going to be made to that nitrogen right there. And again, we'll usually draw the H going straight up. That's not because that's the exact actual geometry. Obviously, we're trying to draw in 2 dimensions a 3-dimensional object. It's just to make this thing not get really ugly on the page. That is a peptide linkage or a peptide bond. Unlike sugars, there's no oxygen left over in the chain. Do we see that? It's just one kind of flowing right into the next. The carbon of one amino acid bonded right to the nitrogen of the next amino acid. And could this happen again? Sure. You throw another amino acid here, you've got the OH group there, the H from the next amino acid, and it can pop on and just keep going. Does that make sense? You can just keep going, keep going, keep going. This can happen in perpetuity. Would a couple labels here be helpful? Call this amino acid 1. We'll call this amino acid 2. We'll call this the reaction site. We've labeled that peptide bond, and we have here the formation of what we would begin to call a polypeptide. Now, I guess that's not really true yet because so far we only have two, but it will form a polypeptide. We don't really get dipeptides like we get disaccharides. I mean, that's possible. You could absolutely stop at two. But in practice, proteins are big. There are a lot of amino acids stuck together. And what kind of reaction is this again? Because water is released. Does anyone remember what we call the reactions where water is released? Yeah, very good. Okay, now, remember we said with sugars, form is function. Form is half of function. Form and chemical properties determine function. So first, let's talk form. So the first thing I want to note is this. Looking at this molecule, is there another site where branching could reasonably happen? And you might be tempted to think that we could branch at this N, have this N bonding to two oxygens, but the geometry doesn't work. It's too tight. If you build it, you'll see there's just not enough space there for that to happen functionally. So we cannot get branched chains with a polypeptide. There's only room for one bond. On an amino acid, which I am going to very cleverly write as AA. Which means what? Which means these chains are always going to be I don't want to say perfectly, that's on, are only straight chains, no branches. Shut up. Sorry. Normally I put my phone on do not disturb, but my son's been a little ill, and so I'm just keeping it on so I can sort of follow that. Though in this case, it was not. Okay, so we said form is function. We said shape determines how a molecule works. And then I told you that proteins, which are big groups of amino acids, are the doingest molecules in our cells, which isn't even a word, but there you go, I used it. So that seems like a contradiction, doesn't it? How can proteins be the ones that are doing all the doing if all you can do with them is make a big straight chain? What I would propose to you is this. Has anyone ever bought a cheap pair of headphones, like earbuds, like the ones with cables? Mostly you people use wireless earbuds now. But you ever had that? What happens the first time you put those things in your pocket for the day, and then you go get them out to use them? Yeah, big tangled mess, right? Big long filaments left to themselves. Anyone ever done a bad job packing their Christmas lights up if they celebrate Christmas at the end of the year, and then gone to retrieve them the next year? What's that like? Big tangled mess, right? And is it a big straight line tangled mess? No, it forms a 3D shape, right? Like they turn into this big, stupid, massive ball that you're desperately trying to untangle, and then Yeah. And if you're like, I think most families, you probably throw them out and buy new ones. Or if you're like my family, because I'm a horrendous cheapskate, it can become like a 4-day project to disentangle the Christmas lights. With them going, just go buy new ones, and be like, no, I'm using these ones. Anyway, this hypothetically. Proteins are like Christmas lights. They have form, and form is function. You see, when we talk about proteins, we talk about their primary structure, and we'll talk about what that means. This is the order of amino acids in the polypeptide chain. All right. And so, for example, if we were building a protein, we might code for it to be built with a glycine, then another glycine, then a methionine, then a phenylalanine, and so on and so forth and so on. That's just describing the chain. And we can actually imagine that in each case we have like a different amino acid. I'm going to use squares. I don't know why I'm going to use squares, but for some reason that's appealing to me. And then methionine, what color is it going to be? It's going to be green. And then phenylalanine, what color is it? It's going to be blue.
Oh, but I know we don't have as many colors as you.
You'll be all right. You're tough. You could always just highlight it later and change the colors. All right, there we go. So, and obviously, when we talk about this, we're really talking about the R groups, right? Because every one of these is connected to an amino acid that actually looks the same, right? Like blah, 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 and so on and so forth. Only the R groups are different, right? And the bonding doesn't happen between R groups, right? If we remember, it is the amine side of an amino acid bonding to the acid side of an amino acid that makes the peptide bond, that makes the peptide linkage. They're the part that are connected. The R groups sit below them, right? I mean, below. Oriented any way you want. I have to draw above here, which I guess is silly, but there we are. It's only the R groups that are different. But the R groups are very different from one another. Some of them are big and bulky. They take up lots of space. Some of them are very small. Some are polar. Some are non-polar. Some are acidic. Some are basic. Some have charges. Some don't. They have different properties. And what are all those things going to do? If you put 3 big bulky ones next to each other, are they going to sit and still continue to make a perfectly flat chain, even though the group coming out the top is big, massive, and takes up a lot of space? Or are they going to push against each other? They're going to push against each other. If they push against each other, what's going to happen to the chain? It's going to, not necessarily tangle, but it's going to change direction, right? It's going to curve. It's going to change shape. So the primary structure refers just to the order of amino acids that we put them in. This is essentially a one-dimensional shape, right? It's just a line. Wow. I'm having a moment. No H in dimensional, that's for sure. There we are. I think I was already thinking about the word shape. At least that's my excuse. Shut up, Ryan. You're supposed to not make fun of me. That was the whole point of me sitting here. And building a protein is taking that one-dimensional shape and turning it into a 3-dimensional shape. So how's that happen? Well, I'm glad you asked.
Did it say a straight line?
Yep. The bond angles of amino acids, their sizes, chemical properties, and interactions cause tangled shapes. Highly scientific language. There are two very common ones, though lots of less common ones. And they are the alpha helix, alpha helices, and the beta pleated sheet. It should have just says it's pleated sheet, but I was always taught beta pleated sheet, so I'm going to throw that in there. These can be either coiled. That looks like called. So if you look here, they're showing that if you put the right groups together in a chain, you can get forces of attraction to form between them. And so certain sequences of amino acids will result in that chain going from being a straight chain to naturally curving in a helix, similar to the shape of DNA. Totally different molecule than DNA, but a similar shape to it. DNA, of course, is a double helix, right? So two of those interacting. If you do a different orientation, you can get this kind of flat section called a pleated sheet. This is just two examples of secondary structure, all right? There's many, many other shapes you can get by putting together different combinations of R groups, but these ones pop up a lot. If you look at chemical structures of proteins, you'll see sections where instead of drawing all of the individual amino acids, they just throw a coil in there. Like that's a section of alpha helix. Or they just throw a little pleated flat space in there, in which case they say that section there. These are combinations that show up again and again, and they have certain chemical properties. But again, any different number of amino acids put together are going to give you a different secondary structure, a different kind of little shape. Does that make sense?
Helix or helix?
Helix is singular, helices is plural.
So it's supposed to be C?
Yeah, but only when it's plural. That's CES, helices. And then if it's singular, it's helix. You'll often see people pluralize it as helixes now, but that's new. And as you know, I'm afraid of new things. They scare me, they intimidate me, make me feel old. Yes? Yeah, the primary structure results in the secondary structure. Whatever that chain is going to result in it folding in a different way. So these are small little local folds, and then the tertiary structure is the protein's individual shapes, which remember are the secondary structures, will all fold together to form a single three-dimensional mass. This is what we call the tertiary structure. That is a word I hate. But this is the working structure of a single polypeptide. And because it's now working, we call this a protein. And all the stuff that does anything in your cells, that is made by your cells, is a protein. You make a big long chain of amino acids. Because of their chemical and physical properties, they fold together into small local shapes. All of those small local shapes fold together to make one big massive molecule. And that is a single molecule. It's actually a single chain. If you could disentangle it, like your big mass of Christmas lights, this could be stretched out back into a single chain. But it is incredibly folded in on itself. It's a big convoluted shape. Imagine if you got, because some amino acids, some proteins have millions of atoms in them. So imagine if you had a string of millions of Christmas lights and you let that get tangled. It could make a really big, complicated shape. That's what we're talking about here. Does that make sense? So primary is just the straight chain. Secondary structure is all those local shapes. Again, we use the two common examples of alpha helices and beta pleated sheets, but you could also have just like a big clumpy spot or a big ball, or you could have a section where it is pretty linear. All the local shapes are the secondary structure, and then how all of those fold together give you the tertiary structure, and that's the working structure of a single polypeptide. That can be a protein. And yet, we go one beyond that, because what comes after that? And that is the quaternary structure, which I meant to put a gap for this, but that's okay. Some proteins, not all, but some proteins are more than one chain put together.
What's your opinion on the word quaternary?
I don't like it. Because I find it very difficult to say. Amino acid chain put together. So if you put two tertiary structures together, they will form a quaternary structure. Yeah. Zoom in comes up. Yep, you bet. Sorry about that. I'm being messy today, and I don't mean to be. I apologize. I'm going to leave. Can you still read that? Because I want to, while you do that, I want to pull up mole view and show you something. Nope, not MoView. Mole view. So if we looked up a protein, and I'm going to look up hemoglobin, because that's like the go-to example. All right. If you look here at this particular protein, you can see that there's some sections where they've drawn like the individual atoms. Those are individual amino acids. But for the most part, everything can be run down to sections that are either pleated sheets, which they show in white here, or the alpha helices, which they show in magenta. It doesn't look very magenta-y up on that screen, but on mine it sure does.
Oh wow, it looks very magenta.
There are different ways that, oh, for God's sakes. There are different ways that they sometimes show this. There's what they call the cylinder and plate model, which still is getting to the idea that you have these bulkier sections and these flatter sections. But by and large, the most common representation is what we call the ribbon representation of proteins. It's actually very hard to find one where they will just show you that. But what I wanted to show you here is that hemoglobin is actually the same polypeptide put together 4 times. That just happens to be how it's made. It's 4 subunits. And so each of these is an individual chain. And so each of these has a primary structure, an order of molecules, secondary structure, where it flattens into beta-pleated sheets or coils into alpha helices, and then some other things. Tertiary structure, where it folds into this 3D shape, and then the 4 of them are put together to make the final quaternary structure of the overall molecule. Does that make sense? I'm going to try to race through this so I can. And there's not much to say. Somehow this ended up on separate pages. I do not know what happened here. I swear I'm having weird printer moments today.
Blame photocopiers.
But changes. Proteins regularly change their shape, and they fall into 2 basic categories. The first is conformational changes. These are designed shape changes to do a job. This is the function of many proteins. Many proteins are designed to change shape as they do the job. Not all of them, but many of them. There are enzymes whose job is to grab a hold of one molecule, grab a hold of another molecule, and then move them into place to each other so they react. Literally like a little factory line assembly, and the protein changes shape. It does that. There are proteins that act as gates that will open and close to allow things into and out of a cell, or to close so that things can't get in and out of a cell. They're designed to change shape. That is their function. And as you can see, those 2 points definitely took an entire page. So now we're going to go to the next page, where the 2 points here are also definitely going to take a whole page. What I'm saying is this note is a hot mess, and I'm sorry. Don't worry, I'll go back to it, but let me just get this here. Some changes in shape are not by design. And inhibit protein function, which is a fancy way of saying they stop them from working. What are some things that can make a protein not function properly? What environmental factors could cause this to happen? Yeah, radiation, damage, sure. What else? That's a good one. Fever. You get a fever. Your proteins change shape. We call this denaturation. And if you get a fever, what your body is doing is it's banking on the fact that its proteins are tougher than the bacterial proteins inside of you. And they heat your body up so that the proteins inside the bacteria denature and the bacterium stops working, and that can kill it. But along the way, it also makes your proteins not work very well. So it's not pleasant for you. And if a fever lasts too long, it can do damage. And if a fever gets too hot, it can do damage. And if it isn't controlled, it can kill you. So that's your body playing chicken with a bunch of bacteria, saying, I bet we can melt your proteins before ours melt. And before the advent of modern medicine, sometimes that worked and sometimes it didn't, and people died. Yeah.
What if when you're like really, really cold?
That will not tend to that will tend not to cause a change in shape, though that will inhibit design shape changes, if that makes sense. So it's still bad. Our proteins are designed to operate in a very narrow temperature window. The human body, and the more sensitive the protein, the more narrow the window. So our organs, the proteins in them that are doing complex jobs, they have a very narrow temperature range they're successful in. You go down by a couple of degrees, we call that hypothermia, and it can kill you. You go up by a few degrees, and we call that a deadly fever, and it can kill you. We're very sensitive. You may not know this about yourself, Preston. You're very sensitive. That's got to be true. Hey guys, have a great day. I'll put a note up what the quiz is about. It's honestly not a deadly one. And if I forget to do that, then you all get perfect.